Good afternoon. It's top of the hour here in Charleston. So, welcome to this edition of New Frontiers in Spatial Omics. I'm Peggy Angel,
And I'm Kristen Burnham Johnson.
and we're delighted to have you here today. So, today's topic is spatial analysis of brain glycosylation. We're honored to have two excellent scientists, Dr. Carlita Lobrilla, who's a world-renowned expert in glycobiology and glycoanalytics, and Dr. Ramon Sun, who directs a new center of spatial metabolomics at the University of Florida.
And then to give you guys backgrounds on how we're going to run our agenda today, we'll have two presentations from our speakers, followed by an interview and then audience discussion.
Okay, so our first speaker today is Dr. Carlito Lobrilla. He's a distinguished professor of chemistry, biochemistry, and molecular medicine at the University of California, Davis. He is a prolific scientist and chemist with over 500 publications focusing on using mass spectrometry to decipher glycosylation, disease, bioactive foods, and gas phase chemistries. He's received numerous awards, beginning as a postdoctoral fellow with the NATO NSF Postdoctoral Award to the recent ACS Field and Franklin Medal for outstanding contribution in mass spectrometry. From his background, he has also founded 3 companies, Evolve Biosystems, Interven, and BCD Biosciences. Dr. Lobrilla has mentored over 70 PhD students and over 200 undergraduate researchers. He was recently elected as Vice President for Programs for the American Society of Mass Spectrometry. Dr. Lobrilla?
Okay, I'm sorry, I had to rearrange something and it, of course, went at the wrong possible moment. Sorry, let me. Okay. All right, so today I'm going to talk to you. Well, first of all, good morning from UC Davis. And I'm going to talk to you about some glycoproteomic and glycomic methods we've had for probing what we call the glycocalyx of the brain. Okay, so as many of you may know, the brain is highly glycosylated. And it's, all right. So the brain is highly glycosylated. And what we wanted to do was really to look at whether we could get some information on the brain and how that could affect what we really wanted to do, which was develop a biomarker for Alzheimer's, right? And so the question then is that if the brain is highly glycosylated, by mapping it out, can we find something in common with blood that would allow us then to find more effective biomarkers? And it's well known that there's already proteins that are involved that are found both in the brain and in the blood. So proteins such as beta amyloid, particularly the apolipoproteins such as ApoE, ApoC, and ApoA. But there's another aspect to this, and that is the glycosylation. So a lot of these proteins are glycosylated. The brain is highly glycosylated. And so we also wanted to know whether there are glycans that we can look at in the brain that could somehow be affected in the blood itself. And so over the years, we've developed methods that we call a comprehensive multiglycomic characterization, and particularly of the cell membrane itself. So we could take any cell lines or any tissue and look at the glycans in the cell membrane. And this will include all kinds of different glycoconjugates. So in glycoproteins, we'll look at the N-glycans and the O-glycans. And in we'll look at also then glycoproteins and then also glycolipids. And the platform has to be quantitative. It has to be highly reproducible. And we want extensive analyte characterization. So this is the method that we've published. We can look at any tissue We can lyse it. We'll do a series of ultracentrifugation to pull out the cell membrane, and then we can get the different fractions from it. So, for example, we could take the membrane, we could release the N-glycan, enrich for the N-glycans, and then do a glycomic profile. From that same fraction, what's the remainder of the pellet, we can do a false extraction and then look at the glycolipids themselves. And then once we have the glycoproteins where we've taken out the N-glycans, we can do a beta elimination and then get an O-glycan profile. The other half of this, we could take what's left over of the glycoprotein, do a triptych digestion, and then we can do a glycopeptide analysis and also a proteomic, so a glycoproteomic and a proteomic analysis, and then get you site-specific glycosylation and protein mapping. So to accomplish this, we need different types of mass spectrometers. To do glycoproteomics, for example, we use the Orbitrap. To do glycomics, we'll use the QTOF or the ChipTOF. And to do quantitation, we'll then use the triple quad. So all of these different instruments are necessary. So the kind of information you get would look something like this. This is the N-glycans, for example, looking at these different cell lines. This is the N-glycomic profile, and there's about 300 structures here. This is the O-glycomic profile, and there's about 50 structures. These are the glycolipids, and there could be a couple of hundred glycolipids here. And so the kinds of things that you would observe, for example, if you're looking at cell lines of breast cancer patients, you would see that, for example, we found that in cancer patients, the high mannose type glycans that come out early here are elevated. And so we've used that as a potential marker for cancer. The other thing then is that if you look at the glycoproteomics of this, you can look at literally see how protein glycosylation change as the cell itself changes or amongst different cells. So here, for example, is a Li cadherin on a CACO2 in the undifferentiated state. We see that these sites are glycosylated with their individual glycans. And then in the differentiated form of the cells, you see that you have the different sites that are glycosylated with different glycans. So we can monitor with with very good detail what the glycosylation is. We can also monitor changes in glycosylation, and we can induce changes in glycosylation. So this is a cell line. If you look at just the native cell line itself, you get the different glycoform. You can inhibit sialic acid, for example. This cell line has very little fucose. If you add fucose to the broth, you can add more, you can get fucosylation. If you inhibit sialation and add fucosylation, you can make fucosylate-only species. And if you add kifoninsin, then you get primarily high mannose. And this is what the glycomic profile would look like. So we can change the glycosylation quite easily. And so one of the things that we want to know is what happens when you change the glycosylation. So you can look at, for example, how the spike protein of COVID binds to different cell lines as you change the glycosylation. And in this case, what we found is that when you desilate it, you can decrease the binding of the S protein. And so you could see the really definitive effects that the glycosylation is really important in this case for binding pathogens. Another pathogen that we looked at, for example, is Salmonella. We found that fucosylation is preferred when you are in adhesion. And then, however, in invasion, you want a high mannose surface. So again, you could see that we've altered the glycosylation here. And we're starting to now see what the different glycan function or what the different glycoforms really mean in terms of their function. So with respect to the brain, we were looking at an Alzheimer's model for the brain, or a cell line model for the brain. And so our collaborators, Azumi Meizawa and Li Wei Jin, created this the cell line model by inducing a pluripotent stem cell to a microglia. And then we wanted to see, for example, what happens with all the different genes with regard to glycosylation. And one of the things that stood out was that the FUT8 which is a gene responsible for producing core fucose, was actually altered as we went from control to the Alzheimer's disease model. So this was a strong indication for us that indeed glycosylation is being affected by um, uh, Alzheimer's itself. And so then we did a more thorough mapping of the brain. And so using our method, we felt it was sensitive enough and with really good specificity to start looking at the different glycomics and glycolipidomics and glycoproteomics of the brain itself. So this, for example, is the regions. We decided to break it up into 11 functional regions. There's, of course, the cortex, the frontal cortex, and so on. And some of these are associated with Alzheimer's. And then the other regions that we've also then analyzed. We had 4 subjects, a 72-year-old with no cognitive impairment, a 95-year-old with hippocampal sclerosis, and then 2 Alzheimer's patients. The N-glycans, and the beginning of this project was started by Dr. Jennifer Tana, who now works for Johnson & Johnson in the Bay Area. And a 40-year student, Ryan Schindler, looked at the glycolipids, and then Yasmin Buchipti, another 40-year student, who she's looked at the O-glycomics. I won't present that data today. We'll focus instead on the glycomics because even there's a lot of work to do. Also, I want to point out my collaborators in this case. I've mentioned Dr. Li Wei Jin, Dr. Izume Meizawa, and then Dr. Angela Zivkovic at nutrition, and then Dr. Daniel Harvey in biostatistics. So this is what the specific analysis of the brain would look like. We separate out the N-glycans, the glycolipids, and the O-glycans as before, again, using different types of mass analyzer. For the N-glycan, this is what we looked at. The little dots here represent the areas of the brain that we actually analyzed. Again, we could get the N-glycome, we get the glycolipid, we get the glycopeptide, and then we can start looking at then the different glycans. So this sort of summarizes the results that we got. So you can see here the different region. You can see that there are changes that are occurring, and you could see that that the changes are actually quite large. As my biostatisticians colleague tell me, if you can eyeball it, you don't need statistics. But certainly we did some statistics. But you can see here that indeed the changes are quite large. And so if you separate these to what people generally study for Alzheimer's, the frontal cortex, the orbital frontal cortex, and the temporal cortex, you could see the changes occurring there. And then there are regions that are not typically studied in Alzheimer's, but we're seeing changes there also. And other regions that also are not studied. And I'll go over each of these separately. And so this is sort of the summary of the glycomic profile in terms of just the classes or the types of compounds. So remember, I told you that fucosylation is important. And you could see here, so this is the legend here. These are the different regions. It's color-coded. But you could see that there are some similarities, but also some very big differences. For example, Pons here has very little of these fucosylation, which is this represented here by the score fucosylation. And again, this is just a representation. There are dozens, maybe even 100 different types of compounds that may look like this. This is the total silylation. This is the high-mannose glycan. And then these are those that contain neither fucose nor sialic acid. But again, you could see the regional variation. One of the things that really surprised us initially was that we always think of the brain as being highly silated. In fact, we did see a lot of silated compounds. But what also struck me was the fact that it was highly fucosylated, and the fucose itself is usually not studied because we focus so much on the sialic acid. But our contention is that the fucose itself is probably very, very important in this regard. One of the other things that we looked at was a degree of branching. So if you compare the brain glycans to those in serum, serum are typically biantennary, but in the brain you get much more triantennary and tetraantennary in very large abundances. And that makes a lot of sense because if you want proteins to talk to each other, the more antennas they have, then the more you get protein-protein interaction. So in the brain, since it's highly interactive, that's the kind of structures that you would expect. So let's look at then the different regions. So the frontal cortex, we see that this is altered by Alzheimer's disease. So this, just to remind you the function, this is performance of motor tasks, judgment, abstract thinking, creativity, and then maintaining social appropriateness. And of course, this is one of the main regions that's studied. And so if you look at this, this is the 70-year-old without Alzheimer and the 74-year-old with Alzheimer's. You could see, again, just visually how different they are. And so here again, you see that there's a large decrease of these highly branched structures compared to not the control, which have these very, branch structures that are highly sialated and also highly fucosylated. And so those are the major differences you see. And if you sort of group them all, so here, the blue here is the sialofucosylated. You see that the sialofucosylated has decreased on a compound level, but also on a group level. And that the green here is the high mannose, and that you're actually producing more high mannose compounds. If you look at it in the individual compounds and you do the statistics, you find that these types of glycans are increasing and these types of glycans are decreasing. Again, you could see that there's a lot of sialation, right? So the sialation has decreased, and also a lot of fucosylation, and those have decreased also. But in this case, again, one of the things that surprised us was not the high level of sialation necessarily, but also then the high level of fucosylation. And so you see that in the frontal cortex, that these types of structures, and these are all statistically significant. We can then look at the temporal cortex, and this is for interpreting sounds, understanding languages, and it's also affected. Here, the changes are not so dramatic, but there are large changes between the individual compounds themselves and within the types of compounds. So, for example, here, sialofucosylated is also decreased in favor of the mannose compound. So we're seeing very similar trends across these two different brain regions. The occipital cortex is not usually thought to be believed to be affected by Alzheimer's, but on the other hand, it's a visual and visual interpretation. But we do see glycans that are affected. And so, for example, if you compare the 70-year-old, 72 and 74, you see that, again, there's large changes that are observed here. In the 90-year-old, you see large changes that are observed here. And now, the really interesting thing that we find here is that if you look at the 70-year-old to the 90-year-old, there's a big change in terms of the glycomic profile that's associated with aging. On the other hand, if you compare the 70-year-old and the 90-year-old with Alzheimer's, there's very minor changes. So in other words, there's a certain phenotype in the glycomic profile of the Alzheimer's that seems to not be related to age. This is really fascinating. And in fact, this is what you observe, that Alzheimer's phenotype is quite distinct, and it's distinct from the control, which tends to vary with ages. And they've seen this with even just looking at the physical brain itself. And we've also, of course, we can quantitate this, and we can see here then the changes that are also associated with age, as well as those that are associated with Alzheimer's. And again, what's sort of interesting here is that the Alzheimer's patient always look very similar, whereas the age in the control will vary according to age, and that just sort of points this out. And of course, we can do, because the method itself is pretty quantitative, we can look at each individual compound and give you an idea of which compound themselves, and it's going into the weeds here. You could really see then the very specific changes that are involved in a lot of these analyses. You could see all the changes of all the glycopeptides and all the glycans according to the different regions, and you can create a clustering analysis here, and you can cluster them according to their age, and again, there are certain things that really stand out for Alzheimer's. Okay, so we also did the glycoproteomic of these in the different region, and so so here, there's so much data that it's actually difficult to figure out what the changes are. You could, of course, do a volcano plot, but what we decided to do was look at, can we observe changes in specific proteins that's related to the Alzheimer's, and also the different sites or the different regions of the brain. And so this is a a protein that we found common to all the different brain regions. And so it has 3 sites of glycosylation. We monitored those sites, and we could see that at the different region, we get variations with the region, but you can also see variations with Alzheimer's and then the control. So you can also then dig down this deeply, and if we were doing a biomarker study, then this is something that we could do. Obviously, with 4 patients, you can't really do an extensive biomarker, but I'll show you how we use this later on. And again, this is another protein. This is ATP beta 3, ATP1 beta 3. So this is the beta subunit of that complex, and it has you can see this as a single site of glycosylation and the glycan types, and then different regions of the brain, and then the different age, and you can see that there. So so this is then the extent of the glycan mapping. There's a lot of data. There's a lot more we could suss out of it. But so far, what we're seeing, of course, is that we're seeing major changes in the glycans themselves. And so now we go to the blood. And so what we wanted to do was then, we know that changes are occurring in the brain. Can we then look at the blood and see if the changes are actually occurring there also? And so for this, we use a method that we've used for a couple of years now. This is, we'll take the serum glycoprotein, produce the glycopeptide, and then we'll do dynamic MRM on the triple quad and look at each individual glycopeptides separately. In this case, I've shown you earlier how we do the glycan and the glycomic, and now what we'll do is we're going to look at the glycopeptide, and I'm going to go through this quickly because I'm probably running out of time. But anyway, what we do is we quantitate this with regard to a specific peptide on the protein, so that we do the glycopeptide versus the protein. And so we're actually changing extending glycosylation. And a lot of this method is now high throughput, so we use this a lot for doing biomarker discovery. And so this is, for example, looking at the tryptic digestion. We're doing the dynamic MRM. So we know that it's linear. This is highly quantitative. We can look at several transitions, looking at the glycopeptide, their abundances, and monitor that, right? And this is, for example, if you look at, we look at many glycoproteins. This is just for what would you would see if you're looking at the immunoglobulins. You could see the peptides that we use for quantitation, and then we can look at the glycopeptides. So the glycopeptide abundances are normalized to the peptide, the non-glycosylated peptide abundances, and in that way we can then quantitate and monitor changes in glycosylation. And so in this, we looked at a cohort looking at, for example, it was 2 cohorts, about 100 controls and almost 100 Alzheimer's patients. And so you do the volcano plot, and what we find in are very specific biomarkers in this case. So you can monitor each individual glycopeptide and see how that would change. And so some of the things that I'll just quickly go to the results here. What we found are biomarkers for sex. So there are certain glycopeptides that you can distinguish between male and female. There are also certain glycans, glycopeptides, that have a much stronger response for one sex over the other. So for example, this glycopeptide here, associated with this protein and this site with this glycan, was a pretty good marker for Alzheimer's in women in this case. However, when we looked at the men, we found that it was not a very good marker. So we're starting to see male and female specific biomarkers, which you would expect, but people don't generally look for. The other thing that we found is that we can now distinguish. So this was a very ethnically diverse group of patients. And we find, for example, a biomarker that was effective for African Americans, but perhaps not for Hispanics and not for Caucasians. So again, just sort of shows you the power of this kind of thing. Okay, and so with that, my conclusion is that I've shown you the map of the brain and how we use it to sort of evaluate and look for potential markers for Alzheimer's. We also find, of course, that the glycans are a rich source of biomarkers for Alzheimer's, and we find that biomarkers in AD will differ for races and sex. And so we'd of course love to get much more brain samples, but that's very difficult to get. And so that's why we transition into the serum itself. So this is my group more recently, and not even that, but and I'd like to thank you all for your attention.
Thank you so much, Carlito, for the fantastic talk. So we're going to go to our second talk, and then we're going to have all of our questions for our speakers after that. It's my pleasure to introduce Dr. Ramon Sun. He is an Anne and Oscar Lackard Endowed Chair and Associate Professor of Biochemistry and Molecular Biology at the University of Florida. Dr. Sun has developed a number of new mass spectrometry-based methodologies to profile and image complex carbohydrate molecules with single-cell sensitivity and fine spatial resolution to assess microenvironmental influences with novel machine learning software. Dr. Sun's current focus is on spatial metabolic etiologies of pulmonary fibrosis and Alzheimer's disease, in addition to lung cancer. He has over 50 manuscript and well-respected journals such as Cell Metabolism, Nature Calm, Science Advances, and Molecular Cancer. And in addition, Dr. Sun directs the newly established Center for Spatial Biomolecule Research at University of Florida. And with that, his presentation today will be leveraging functional spatial metabolomics for hypothesis-driven research in the area of Alzheimer's disease and related dementia. So take it away.
All right. Hey, thank you, Kristen, for that. And thanks everyone for joining to this webinar. And I'm pretty excited to share some of the latest unpublished work from our lab, really focusing on multi-imaging technologies. So just the brief introduction on why we care about spatial and why we need spatial technologies to continue advancing from that perspective. So when I was a graduate student, we did a lot of what I call blending specific techniques, where you put tissues of interest, put in a literal or metaphoric grinder, and you kind of get genomics information, proteomics information, and metabolomics information out. And so these techniques are still extremely useful, but there are definitely barriers for this. For example, there are examples of false positives and false negatives when you start pulling a tissue together. So on the right, if you can see, this is a piece of tumor that's no bigger than the size of an M&M on a microscope slide. And this tumor would consist of multiple different regions, just to name a few, like tumor, stromal, necrosis. And it is highly likely that all the different regions in this piece of tissue have different metabolism, right? So imagine if you care about what the tumor phenotype is, but you're blending many different cell populations with different levels of metabolite, what happens when you get a net negative, right? So I call these, really barriers in research. I call these false negatives. So when we see things that doesn't change, and as a scientist, we tend to ignore it. But when you actually break it down, if things do end up changing on a cellular or spatial level, we would never be able to see it. So because of this, we're really pushing the field of spatial biology, which is pretty much has been around for about a decade, right? So from DNA, RNA, protein to metabolites, there has been exceptional and amazing advances in all of these fields. So DNA and RNA, we have single-cell RNA-seq, and we have Visium, and now we have single-cell spatial RNA-seq. For protein, we have spatial proteomics such as Codex, BBTOF, and CyTOF, and a lot of that came from the Stanford, empires over there that really helped us understand the cellular interactome and neighborhoods inside of complex and terminal diseases. And now there's also spatial metabolomics, which is what my lab really studies, that we look at spatial metabolites and how they're co-localized in different disease, in different cell layers, as well as how they change in different diseases. And I'm glad that we're not the only people who's really interested in all of these, because Nature just reviewed not long ago. Nature just published the 7 technologies to watch in 2023, and single cell metabolomics is one of the ones that's highlighted along with the new space microscope. So, we're in a really cool upcoming field that is, just ripe for new technological advances and method development. So the way that we do spatial metabolomics is really using a what we call a multi-mass spectrometry imaging methodologies. So what we can see here in this video, this little bright spot is it's a high power laser that samples through a piece of tissue which is behind through an XY coordinate. And as the laser shoot, we're getting the data collected from all the different pixels that is shot on this tissue. And once everything's done, we computationally combine all of the different pixels as well as their metabolite levels or MZ features and height and through high dimensionality reduction, we can get beautiful spatial distributions of all the different kinds of metabolites. And so the top one, this is a non-small cell lung cancer model that we study frequently in the lab, and you can see that this one unique lipid at very heterogeneously localized around different part of the lung cancer as well as different normal lung. With that, I just want to give a little advertising slide that the new Center for Advanced Spatial Biomolecular Research that's open here at the University of Florida. We have two of these high speed timsTOF multi-imaging instruments. And when you look at it from the door, they look like two thumbs up, which is always bring a smile to my face when I walk into the lab. But also we also have the COIA imaging instruments to study histology as well as cellular phenotypes that help boost the spatial metabolomics analyses. And finally, we have a hydrogen deuterium exchange, which is also spatial, but aimed to look at protein ligand binding on a very molecular level, so not on the tissue level. But I'm not going to go into that for today. Today, I'm still mainly going to go through the multi-imaging of tissues. So the current field or the technology stands at about 5 to 75 micron resolution for each of the multi laser shots. It's single cell resolution because usually cells are less than 20 to 50 microns. But because the way that the XY coordinate actually samples across, it is possible to hit multiple cells in a single pixel. So we're coming close to near single cell resolution, but we're not there yet from a technological perspective. But it is really good at in mammalian tissues to pick out cellular tissue layers where multiple different type of cells like kind of push together. So it's really good to study cellular neighborhood analysis. So with the power of multi-imaging analysis comes the biological question, what we need to ask, right? So when you have, say, for example, a microscope slide with cells on top of it, you've got to understand the cellular metabolic landscape is not just metabolomics, glycomics, or lipidomics, right? It's a interconnected network of metabolic pathways encompassing all of the small molecules you can see, encompassing all of the lipids you can see, and encompassing all of the glycans you can see. And from my perspective, so I'm a biologist, a molecular biologist by training. So the way I would think this is that if we're only looking at a single modality, we're only looking at the pictures through a very narrow lens. So you never get the full picture of what exactly is happening from a disease to a non-disease setting. So essentially, if we don't get all of the interconnecting metabolic pathways from that cell or from that region, we're really missing a critical aspect of the different metabolic perturbations within that brain. So with that, that we went ahead and really wanted to develop a workflow that assesses metabolome, lipidome, and complex carbohydrates through a single tissue section. So you can just go ahead and cryosection a single piece of tissue, whether it's brain or the lung, whatever you feel like doing. We just need that single piece of tissue. and I would like to acquire all these different metabolic informations or interconnected metabolic networks from a single pixel, so I can really do pathway enrichment to figure out what it's giving me a better painted picture of what's going on from a molecular level. So with that, we ended up getting this workflow to work. So you know, we can do metabolome, lipidome, and glycome, as well as glycogen from a single piece of tissue. The whole process takes about 72 hours. Can get up to about 1,000 annotatable metabolites, but obviously there's a lot of peaks that we still don't know what they are. So you could get up to tens of thousands of different features. But we like to play around with all the metabolites that we know the MZ values to, so that we can actually use it for pathway enrichment. So when we finished about this workflow, I have another problem, right? Because we all know that multi-imaging analysis is very time consuming when we go flip through one image at a time to figure out what exactly is changing at different regions. Obviously, that's no fun for me, but it's not, and graduate student also hates that too. And if you give them now that you have to look at through 1,000 different images for a single cohort of study, it's going to take them months or even years, well, not years, but months to complete. So because of this, we ended up developing a bioinformatic workflow that we're now putting into them as a software graphic user interface format, where we can take all the different modalities, co-registering them and collapse them in a single metadata file, and then perform correlative and spatial pathway enrichment networks for the analysis. So for example, we can now collapse all the metabolome, lipidome, and glycome to a single working multi-imaging file. And now you can even look at what's co-localized between lipid and metabolites. For example, this lipid on the left and this metabolite on the right have almost identical spatial distributions. And obviously, we think there's some biological connections over there, but we don't know yet. But it's good to see these information, and therefore it will allow us to continue to work on understanding the biology underneath at least the brain, right? Because it's one of the most complex organs out there. But with that, we can also start build networks of different modalities and see how they are regulated, co-regulated through the different brain. And then, similar to single-cell RNA-seq analysis, we've implemented high dimensionality reduction analysis. So we take all of the tens of thousands of multi-pixels and all tens of thousands of features, and we collapse them into a two-dimensional analysis where we can segregate different regions or clusters based on similarities. And from here, because we have the XY coordinate, all the different pixels, we can actually bring all the regions back down into the spatial format. And immediately, I think, you know, for anybody who studies the brain, you can see that you can really fully appreciate that these clusters do not occur by random. They actually look like actual functional regions of a brain. So this is a mouse brain that's cut coronally. And then talking to a pathologist as well as a brain anatomist person, we can actually annotate all the different brain regions. And what's even more surprising is that means metabolism underlines brain physiology, right? So metabolic processes that occurs in the brain really forms the basis of all the different anatomical regions that perform different metabolic functions. And from here, we can now directly pull out from the different clusters and use them for pathway enrichment. And we can kind of see at what different brain region, ideally, what they prefer to perform in terms of metabolic processes. So for example, this hippocampal region, starch and sucrose metabolism and Warburg effect seems to be taking priority compared to the rest of the brain. So with that, so this is kind of where our software aspect of things we're working towards. But also I want to switch gear a little bit and talking about how we can perform multi analysis in 3 dimensions. Because, you know, 2 dimensions is awesome, but we live in a 3D world. It's just naturally, we're gonna have to evolve to figure out what metabolic responses or profiles look like in a 3D format, right? Because nothing is a square, and even within cellular layers, it changes shape in a 3-dimensional space. So to do this, we just went ahead and start cutting a mouse brain 50 microns apart And we started setting up the raster pixel sizes 50 microns apart as well. So we like to call this the brain, the mouse brain at 50 micron resolution. And here's what they look like, right? So you can cut brain at 50 micron resolution. And we thought, okay, let's just combine this and see what we get on a 3-dimensional setting. But immediately we came across a lot of different challenges to be able to render everything in 3D. For example, we have slide variation from abundances issues. MALDI is not the most consistent technology out there. So there needs to be mathematic transformations for between maybe slide number 1 or slide number 80 that's run maybe 3-4 days later. Also, there's tissue alignment because it depends on how you mount the tissue on the microscope slide. It also needs some level of mathematical or computational work. And also just tissue handling, right? So we have people that just accidentally, mishandle this piece of tissue, then a piece of brain fall out. And how do you handle these kind of analysis in a 3D setting? So because of that, we come with a brand new workflow called MetaVision 3D, and it's available on GitHub now for everybody to try, and it does a lot of different things for the 3D rendering. So let's go through all the different challenges. From the beginning, this is what a couple slices tissue look like. And you can see that immediately this row in the middle seems to just be lower, while the spatial distribution of the lipid looks pretty good, but then it's significantly lower compared to the rest of the other tissues. And I tell you that these are normalized to the tick of each run, right? So because you cannot run everything in one go, we were running different tissues over the span of 2 weeks. So we implemented math, and we used the mean average across different tissue sections. And I think if you look on the left, every 4th slide is different, right? That's from left to the right of the tissue. So we mount 4 different brains from left to right. And it's just funny that every 4th slide seems to be out of whack. So using mathematical transformation, we can bring almost all the brains into a very similar covariant even mean. And then you can bring all of these abundances back up so that you can actually, render your metabolite feed of interest in 3D with a much more accurate setting. So, and then the final, the next biggest issue is actually tissue alignments. How do you align different tissues that, 1, don't even have the same shape, and 2, you're looking at 100 different tissue sections, right? How do you do it in a way that you don't piss off somebody who's the operator who's manually aligned these? How do we do it automatically? So we come up with this called computer vision, which is a AI framed network that uses similar features between different adjacent slices for tissue mapping. And then what I wanted you to see is, so the top one is manu fit. So if you fit every single slice, and you can see that how when it transitions from the first to the last slide, it's jumping up and down all over the place. But if you use our computer vision to apply, you can see that there is no longer that jumping. And I want you to see where the last tissue section is between the 2 different fitted, because it matters, because the brain is a non-linear shape. So towards the end, sorry, I think you guys can see that the tissue position shifts because it's no longer the tissue become much more smaller. Finally, again, we love AI because of the alignment issue. So we figured out another AI method. Essentially, if you have mishandled tissue and you have a giant gap over here, we can take tissue slices from either left or right, and we can estimate the pixel values from the tissue from left and right. And you can see that, and we can fill in this area, we call this imputation, and we also can mask around the regions. And so the top one is before, where the tissue is mishandled, where a piece of tissue fall out, which is pretty standard, by the way. It's common when you section frozen tissues by MALDI. But now look at the bottom, right? You can barely tell that this tissue is actually damaged previously. It looks near perfect to a human eye that AI can perfectly fit this. So with that, we can put everything together, and we can start rendering everything. in 3D space. So now here you look at this lipid that's exclusively enriched in the cerebellum area. And you can look at it in a 3D setting, or you can look at it from a two-dimensional transition and look at what each of the brain region looks like from both of these. And finally, all of our data are currently put up in this website called MetaVision 3D. So you can explore all the different available features here through slide transition, but also you can start rendering your favorite metabolites and play around how it looks like in 3D space. And there's a lot more functionalities. If you just visit the website, we have tutorials on the bottom. And here is the website for how you can do this in 3D. So with that, I think I want to shift gear a little bit on in terms of biology, because that's kind of what the title is. So my lab is actually focused on, complex carbohydrate metabolism in the brain. And we've, published a decent number of papers in the last couple of years, highlighting a lot of the dysregulations of glycans and glycomics in the mouse brain. So, but now we have these, other additional tools in our hands, right? So we have metabolome, lipidome, and glycome. So we apply the SAMI framework on Alzheimer's disease and see what exactly is happening. So the ones I really want to highlight is, so we compared normal and AD brains. So here you can see the cortex, hippocampus, and hypothalamus. And the bottom is small molecule, lipid, glycans. And it's from the left to right is wild type AD mouse, which is 5XFAD, wild type 5XFAD, wild type 5XAD. And to our surprise, I guess not anymore after the first talk, is what you see the degree of change is that the N-link glycans are extremely enriched and increased abundance compared to the other modalities when you mix all of the other combined. So there's definitely changes in metabolome and lipidome as well, but it's just the changes are not as pronounced as it is on the glycan level. And here's a representative example of the glycans from the wild type and 5XFAD mice, where you have all the different pixel by pixel analysis and showing that it's this particular glycan severely upregulated in the cortex, hippocampus, but not as much in the thalamus of the mouse brain. So the next question is, do we phenocopy this in the humans, in the human brain, right? So a good thing lucky for me, I have access to great collaborators in ADRC who provided us with a ton of human AD samples. So the human AD brain, the human brain as a whole is highly variable. If in our experience, brain 1 and brain 2 and brain 3 could have complete different profiles. So it's really hard to power analysis in terms of that. That's why we need a lot of different N numbers to study the glycan spatially. So these are all frontal cortical tissues, and we got about about 5 to 8 per group. So we have age controls, and we also have AD divided by low, medium, high by Braak staging, and you can kind of appreciate that a lot of these glycans are trending higher in the frontal cortical region as the disease is developing more severe. And then here is the data analysis done over here. You can really appreciate the change in glycan abundance as you progress from normal aging to the AD high species to the AD high people who have like really high Braak staging. So with that, we wanted to ask the question is like, why or how is glycosylation triggers? Is that a driver of poor cognitive performance or neurodegeneration, right? Because phenotype, it does not mean, it's just correlation. It does not equal causation. It could be a carrier event that just come around for the ride through some other mechanisms, right? So it may not be a druggable target. So one of the things that we got is really interesting from our study that we think is, so when you make N-link glycans through the ER, trans-Golgi, and finally in the cell membrane, you can do it through the base molecule UDP-gluconate. So you need that to initiate glycan biosynthesis. And you can make this molecule through either glucose through the hexoamine pathway, or you can have glucosamine that directly comes in here through the GLUT2 transporter in the brain. So glucosamine cross the blood-brain barrier and directly taken up into the neurons. And again, we took the AD mouse. So this is just AD mouse, right? There's no wild type here. If you just give the AD mouse glucosamine, and you can see that we can already just inflate what's already high even higher, right? So just assuming the water gavage is already high in these AD mice, but when we give it glucosamine, it became even higher in terms of the end-link glycosylation. And then now the key question is, if we can push glycosylation towards the negative end, or increase hyperglycosylation, what happens to the behavior aspect of the mouse? So first of all, we have water and glucosamine look at Moore's water maze, which is a hippocampal memory test. You can already see that the mouse that has been taking glucosamine is performing much poorer. And then we did what we call a tube dominance test, which is a frontal cortical behavior. So the mouse will push each other through an enclosed tube, and you can record who wins more. And you can and then based on this, you can already see before you gavage glucosamine, there's not much difference between wild type and 5X FAD. But as soon as you start the gavaging the 5X FAD, the AD mice starting to act a lot more aggressive, which is commonly a symptom of Alzheimer's disease patients. So they end up winning a lot more in the social memory, social behavior test. And then we have what we call social memory test. So social memory test is you put these mice together every single day, and you see how well they sniff each other. And on day 5, usually they've seen each other, they will not continue to investigate, right? Because they already know each other. And using the laptop, we can put AI into this as well. So you can kind of see this little mouse on day 5. So this is the mouse that has glucosamine. He cannot remember that they've had 5 days of interaction. So he just keeps sniffing this other mice, even on day 5. Well, this other mouse was being with him for 5 days. Is like, look, I don't really want to sniff you, so get out of my way. And then, so here is the graph of the social memory behavior. And this is what happens when you put glucosamine in the cohort, and they significantly compromise their social memory behavior. So why is this important? Why is this impactful? What I really want to get to is Alzheimer's disease progression. It's super variable. You know, you have people that progresses within 2 years and bedridden and rely on a breathing apparatus. Or you have other Alzheimer's disease patients as you get diagnosed, 10 years later, still walking around in the shopping mall, memory is not so good, but still fully lucid and cognitively okay, even though they have been diagnosed with Alzheimer's disease. So there's a huge variability. And what I want to get to is, we don't know what we put in our body that will trigger this variability, right? So we don't know anything that we take that would have any effect in the brain. And glucosamine that I'm showing here is one of the most common taken supplement for joint issues in the elderly population, who are the most at risk in Alzheimer's disease. And while we don't have any epidemiology studies, I'm almost willing to go out on the limb and say that a lot of Alzheimer's disease patients are actually taking glucosamine during their course of disease, because they also have other joint pains due to age. So what we found in this study really warrants additional studies in humans as well as mice, really fully figure out, if there's something that we're taking that's modulating our brain, even if it's glucosamine, for example, through the N-glycan pathway, that could be something that we can stop immediately and maybe reverse or delay the progression of Alzheimer's disease for a lot of the people who are suffering from it. And with that, I'd like to close my talk by thanking everybody in my lab, amazing collaborators. And I do want to highlight, Peggy and Rick at MUSC. They pretty much held my hand when I bought my first multi-instrument and showed me all the ins and outs of running that instrument. So I own a great debt of gratitude to them, and also have a lot of other collaborators on the mouse side. Here's my funding. Feel free to check out the Mouse Brain Atlas, and here's the center website. And with that, I'd be more than happy to take any questions.
Thank you, Ramon, for such a fantastic talk. Peggy and Carlito, do you guys want to come online too?
I'm not
Start
able
getting-
to start my video. I don't.
No worries.
Lydia,
They
thank
took all
you so
the
much.
options away from Peggy.
They've taken my power. Thank you guys so much. What a great pair of talks.
Yeah.
Looking at spatial analysis of the brain. Kristen, did you have any questions?
I think for my main question for both of you is really, what do you think is your most significant finding from this? What were you most excited about? Maybe start with Carlito first and then move to Ramon.
I think for me, it was the fact that it's not just all sialic acid. I mean, neuronymic acid gets its name from the fact that it was found in the brain. But it looks like there are other glycans that are, or monosaccharides that are really important. So other types of glycans that are really important. And the other thing is that I think we're starting to see things that perhaps we could use as biomarkers for Alzheimer's. And you know, people used to say that, why do you need a biomarker for Alzheimer's when you couldn't cure it? But now that we have different, now that we have drugs that may, or at least alleviate it, and also if you have a biomarker, you can do a more effective discovery. I mean, you could do a more effective therapeutics. And so it's a chicken and egg thing. You know, if you have biomarkers, then you can actually get better therapeutic. If you have therapeutics, then you need biomarkers. So
Yeah, maybe glycomics has been that missing link to find a lot of these biomarkers that we've been trying to find over the decades.
yeah, absolutely. And the thing about glycomics is that it's highly quantitative. It's a lot easier to quantitate glycans. It's harder to interpret the structures, but it's actually a lot easier to quantitate the glycans. And that's also what's been missing, is that high quantitative analysis.
Wonderful. Ramon, what about you? What's your most significant finding that you were most excited about?
Well, if we have to come down to one, what I really want
You
to
could do a
push
few.
is, what's the few? One, well, there's two different aspects of this, right? Is the new technology enables a lot of new molecular biology and hypothesis-driven research, right? That our research don't stop at the mass spec. We actually, our research starts at the end of the mass spec run, right? It generates so much hypothesis data, so much data to generate hypotheses that we can test in mouse and human models. That what I really want is the actionable, real, actionable data that will impact human lives in a lot. And I'm hoping to do that. And for example, the glucosamine story, you know, we're really, you know, excited about doing that in human studies, is because we honestly don't know, right? We don't, our data seems to suggest N-link glycosylation is dysregulated in neurodegeneration, but you can make it way worse just by taking a simple supplement you buy from Amazon or the mall. And I guarantee you, there's a lot of people who takes it, right? So the glucosamine, if you go to any Amazon link, the most popular glucosamine supplement have over 100,000 reviews. So you can just kind of imagine how many people are actually taking these for their joint. And there are published studies that's showing that these are beneficial for your joint. But what are we doing with these supplements with respect to our cognition, especially at the at-risk population? So, you know, I'm really hoping that once the study is published, we can really do some good to the Alzheimer's disease population, right? So there's plenty of therapeutics being developed for Alzheimer's disease. I go out on a limb and say not a single one of them is actually effective currently. So and I'm going on the route of like smoking for lung cancer. Once we're starting the campaign to eliminate smoking to alleviate lung cancer, I think by estimate, since the initial campaign, the finding has saved more than 400,000 people who would otherwise get lung cancer. So, piggyback of some of the stuff we've done in the cancer field. We feel like just modifying our everyday activities like a simple supplement, I really do hope that we can change the disease progression in Alzheimer's disease patients.
That's fantastic. Thank you.
I have a quick question. Well, I hope it's okay if we go a little bit over, Lydia. So Carlito, so you had some really interesting tissue studies and then blood studies. You had the quaternary Lewis antigen that seemed to be presenting in the Alzheimer's tissue consistently in the 70-year-old and the 90-year-old. Have you seen any connections between serum expression of glycans and tissue expression?
Not as direct as we would like.
Mm,
I think what we discovered with the brain was that there are changes in glycosylation and it is affecting it. So we thought it would be systemic.
mm,
The problem in serum is that a lot of the glycans are biantennary.
mm,
So the tetra and the quarter, the tetra and ternary are actually not very abundant in serum at all. So you really have to dig deep to get those kinds of structures. So we weren't able to see that kind of effect. But some of the things that we wanted to look at was that whether because you have the same enzymes, the same sialic acid transferase and the same fucose transferases in the brain and in the serum. And so we thought perhaps those are the changes that you would observe where you would see changes in fucosylation and silation more generally. So those are the kinds of things that we look for and that we are looking for.
Have either of you looked at any samples related to Down syndrome? They have accelerated Alzheimer's. Probably not.
Funny you mentioned that. We have so not human, but there is a colony of Down syndrome dog that has been used for to study AD for that particular region. So Down syndrome dogs. So we have looked at those brains. I think Liz at UC Davis or Berkeley, UC Berkeley. So yeah, she's over there. So she moved from Kentucky to bring his Down syndrome dog colony over. So when she was in Kentucky, we chatted and we ran some samples, and you kind of see similar patterns of link glycosylation dysregulation in the Alzheimer's disease dogs, Down syndrome dogs, who's a model of Alzheimer's disease in I guess larger primates.
And that is an interesting question, Peggy, in many ways, but also it highlights sort of the fact that we're really all just sort of sample limited.
Exactly.
So I think the tools are there, and hopefully as people start to see what you could do, that they start reaching out to us and start looking at these these problems. Certainly that's happening in other diseases that we've done. But the other thing that I want to point out is that I think it's great. So Peggy selected these two talks, and I think they're great in a way that you could see what they're actually very complementary. And I think using both of them helps you understand a much bigger picture than what we individually show.
Yeah, excellent talks. I think the animal models give some sort of leverage for developing technologies for early detection, and then just finding those valuable samples in the human population is what we need, because we don't want to donate parts of our brain, right?
In that vein, funny enough, we actually looked at oligosaccharides in the milk of cats and dogs. And the weird thing about cats and dogs is that they're actually very similar to human. So cats represent one group of human, like the non-secretors, and dogs represent, their glycans represent another group of humans, the secretors. So I think the dog model is probably pretty good in this regard, or could be.
Interesting. So it looks like we've answered the questions from the audience through text. Kristen, did you have any more questions? Or Ramon, did you have questions for Dr. Labrilia and vice versa?
Mhm.
All right. Thank you guys for this
Yeah.
session. We'll
Thank
see you
you
on our
so
next
much.
webinar. Thank
Yeah.
you.
Thank you all.
Bye.
Bye.
Thank you all. Bye.